Hi everybody, I'm happy to introduce a new eDNA-based method for detecting the presence of an endoparasite in salmonid fish. The myxozoan parasite Tetracapsulides bryosalmoni is causing the proliferative kidney disease and affects the whole range of salmonid species in Europe and North America. During the last 2 decades, it has been responsible for big declines in both wild and hatcheries population. And as its development depends on water quality and temperature, more outbreaks of the disease might be to come. The detection of infected fish usually relies on kidney sampling for detection of parasite spores or DNA. And this means killing the fish to infer population infection prevalence and hinders large screening that are needed to understand this disease dynamics. So to deal with this issue, we have developed a non-lethal method for detecting the parasites in its fish host. Knowing that infected fish release parasite spores in their environment along with their urine, we have put juvenile brown trout in small volumes of non-contaminated water and waited for them to excrete. We expected that an infected fish would release parasite spores or at least parasite DNA with their urine. And after filtration on the field, DNA amplification out of the filter would thus be able to assess fish infection status. We have tested this method on 8 different sites, showing contrasted environmental conditions on a total of 111 fish. When comparing the detection of parasite DNA in the urine with the droplet digital PCR and that detected in the kidney of the same fish, we get 90% agreement between the two methods, showing that uDNA is efficient to detect either positive or negative individuals, and that regardless of their site of origin. Applying this uDNA method, we have been able to draw a map of population infection status on a wider scale. We then tried to link this map to environmental parameters that were measured on the field. On this redundancy analysis, we can see that population infection prevalence, as well as the quantity of DNA excreted per fish, are explained especially by mean summer temperature and to a lesser extent by nutrient quantity in the water. This was expected as temperature increases parasite multiplication and transmission, and together with increased concentration in nutrients, they enhance the growth of bryozoans, which are the parasite's primary hosts, and thus it is increasing the parasite niche available. So thanks to our new method, we can infer population infection prevalence and dig more into the environmental factors that are influencing the interactions in this parasite complex life cycle, while at the same time being able to release the fish back to their stream after excretion.